You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Jacobs with the Future Tech and Finding Genius podcast. I have Isabel Mansuy. She's at the Laboratory of Neuroepignetics. She's a professor in neuroepigenetics at the medical facility of the University of Zurich, Department of Health Science and Technology, also the Swiss Federal Institute of Technology. So Isabel, thank you for coming. How are you doing?
I'm doing good, Jen. Thank you. Yourself?
So I know about epigenetics, I guess, is the access or the changing of the ability of genes to be expressed, expressed more, expressed less. But what is neuroepigenetics?
Neuroepigenetics is the ensemble of epigenetic processes, factors, or mechanisms which exist in nerve cells, in the brain and in the peripheral nervous system. It's basically the same basic mechanism as in other cells or organs or tissues, but we focus essentially on the brain.
I guess methylation and histone, deacetylation, that kind of thing.
Yes, DNA methylation, RNA methylation, histone modifications, acetylation, methylation, phosphorylation, and so on, but also more recently RNA, non-coding RNA.
What circumstances have you observed epigenetic changes to happen, in the neurological sense, and then what gets regulated typically?
Everything. I mean, a cell itself, whether in the brain or whether in other tissue, needs epigenetic factors to function, to be created, to differentiate, to grow, and to function. So there is nothing really special about epigenetic factors or mechanisms in the brain. What's special is the relationship to brain functions. And in the brain, there are many epigenetic marks or factors, mechanisms which exist naturally whenever, if the brain is at rest or whenever it's activated. But there are specific factors which are regulated or which are turned on or off when we learn some new information, for instance, when there is a fear memory or any kind of memory. So there is some specificity about epigenetic mechanism in the brain, but there are many of these mechanisms which are similar as elsewhere in the body. What are some specific areas of your study? Are you looking at people under learning environments, under stress environments? What are you focusing on? I'm really mostly interested in epigenetic inheritance. Actually, my chair is called Neuroepigenetics because I'm a neurobiologist by training. But since now more than 15 years, I'm mostly interested in epigenetic inheritance, which is how life experiences affect our health, whether our mental health or physical health, and how these effects can be transmitted across generations. So this calls on mechanisms which depend on epigenetic factors, because the environment, whether the social environment, what we eat, what we do, physical exercise, and so on, the endocrine disruptors or drugs we are exposed to, they act or they modify the epigenome or epigenetic factors, and they leave traces which affect the brain and sometimes across generations.
All the cells of the body, but these cells that form that are part of our germline are the only ones that can carry forward any epigenetic marks, right?
When we talk about true epigenetic inheritance, yes, the alteration, the changes in epigenetic factors have to be in germ cells. Yes.
Are you saying that, okay, if I experience
terrible anxiety throughout my whole life, let's say, because I have just horrible living conditions. Are you saying that, is there evidence that my children or grandchildren may experience that same thing if they've inherited essentially the anxiety, the effects of the anxiety because it's changed me epigenetically throughout my life, for instance?
Yeah, I mean, there is a lot of evidence that some symptoms or some diseases can be passed from parent to children, sometimes across generations in human. So this is known. What's not known yet in human is whether this is based or this depends on changes in epigenetic factors. So there is correlative evidence in humans, but not causal evidence yet. The evidence exists, but only in experimental animals, in mice, mostly in mice, and some results in rats. So in mice and rats, it's known that if you are exposed to a trauma, if the mice are exposed to trauma in early life, or if they are exposed to, they are fed a high-fat diet, this will alter their health, mental and physical health, their behavior for trauma, and the symptoms will be transmitted to the following generation. And thus, this is causally due to changes in epigenetic factors in the germ cells. Evidence in human is still lacking because, I mean, you imagine that it's not easy to do this type of causal analysis in humans.
Because each cell type in a mouse's body, for instance, would experience an effect differently. But again, it seems to, in terms of inheritance, all come down to the germ cells, how they are affected. So, I mean, it's odd. I guess they're affected directly, but indirectly. They're not affected necessarily,
Yes, you are completely
right. How would you describe it? I'm sorry.
Yes, you are completely right that they may be affected directly or indirectly. I think it depends on the type of exposure and on the time period, the time window of exposure, and on the duration of exposure. You imagine that a young animal or a young human where the gonads, so the testes or the ovaries, are not completely formed yet. They are not as protected as in the adult. There is a blood testis barrier, for instance, in human and in mammals in general. So it's a cellular barrier which protects germ cells. And this barrier is not completely formed in early life. So at birth and in human for a couple of years after birth. So this is a window of time when there is higher sensitivity of the germ cells. And they may be affected directly by blood, for instance, or if the individual is exposed to endocrine disruptors, the endocrine disruptors may cross the blood testis barrier, or what's the forming the testis barrier in formation, and will reach the germ cells, the developing germ cells. The same is true for trauma or stress. There are many hormones, including stress hormones, corticoids, which will be released, and they may reach germ cells and have an effect on these germ cells.
In terms of short-term and long-term effects, let's say, in a rat, how long does it take for a new sperm to form in the mouth?
I mean, in mice, it takes about 35 to 40 days. In human, I think it's a little longer. So it's true that, I mean, the time of if exposure happens early in life, it's likely that the germ cells themselves will be affected. And if the germ cells are affected, it may be that all cells produced from these germ cells will have the alterations. Because
I can see a couple different ways. So you're right. If it happens early in life, every single sperm that's created in the mouse from then on may be affected.
Yeah.
But if you're, let's say, I don't know, a negative stimulus happens to a mouse right before it happens to start forming, a sperm cell that's going to be the one that makes the female mouse pregnant. That could be an effect, and that could maybe happen if it happens in one very short time period, in one day. Or if you even if you have a short-term chronic exposure, let's say it's for a month and a half or a month, and that just happens to be the worst time for it to happen. The mouse could still maybe not be completely changed, but pass on the negative experience.
Absolutely. No, absolutely. There are different levels. There may be a temporary effect of exposure on germ cells. So one offspring generated at some point shortly after exposure may be affected, and offspring generated much later or years in human or months after in mice will not be affected. It's very true. Now, there is also the question of inter versus transgenerational. Depending on the severity of the exposure and on when the germ cells are affected, this may be totally embedded into the germ cells to a point to be transmitted further, not just to the offspring, but also to the grand offspring.
Yeah, like I guess, it's known there was the Dutch starvation winter, during World War II, and I guess the children were documented to have, the epigenetic changes. I don't know about grandchildren, but do you know if that study's gotten to that point?
Yes, yeah, but in this study, it's pregnant women. I mean, some women were pregnant, others the babies were born already. But in the babies who are born, their own germ cells were already exposed, right, in their body. So that their own children have effects can somehow be understood if there was indeed direct exposure. Now the question is whether the further generation where not a single cell, not even the germ cells that gave rise to them, even if it was a germ cell in a child, if it has not been exposed, then if there are symptoms, it means that there is transgenerational inheritance. And that's what we see in our mice, for instance. In our mice, in pups exposed to trauma, some of the symptoms are expressed in the fourth and sometimes even in the fifth generation. So in animals which have not been at all exposed, not even the germ cell they come from has been exposed. So there is a very big difference between intergenerational and transgenerational.
Well, there just seems to be like an adaptation that the cells make to their environment. So have you seen the effects die out after a certain number of generations, depending on the effect?
Yes, indeed. Depends on the effects. In our model, for instance, depression affects the third generation, but it's no longer observed in the fourth generation, while risk-taking is still present in the fifth and a little bit in the sixth generation. Other symptoms, they disappear after the first generation, so there is a little bit of every possibility. I think we don't know why. It depends probably on the type of genes which are recruited for this type of behavior, on their vulnerability or sensitivity, on their likelihood to be affected epigenetically. And it may also have to be with the importance of the behavior itself. Risk-taking is a very kind of primitive type of behavior, which can be both sides. It can be dangerous to be too risk-taking, but at the same time, it can be beneficial to be quick and to be prepared to take risk. So you see, it's a very vital and fundamental type of behavior. It could be why this is particularly affected by environmental factors. I don't know. I'm just speculating here, but it's quite striking to see that risk-taking is this type of behavior which is affected for so long.
At least for DNA, I know DNA can be sequenced, but can you sequence a mouse, for instance, and capture all the epigenetic marks? Is that even part available yet?
Well, we can capture all RNA, we can capture all DNA methylation, but we cannot capture all histone modifications because there are just too many. Even DNA methylation, capture it all, whole genome bisulfite sequencing is required, and it's almost 100%. But then, what we do from this data, there is quite inter-individual variability. And intercellular variability. If you take two liver cells next to each other, they will have slightly different DNA methylation profile. So what to conclude from, even if we are able to capture all these epigenetic factors, how to link this one or that one to the behavior or to a particular symptom is very complicated. It's just impossible for the moment.
Well, what kind of stress do you put the mice under to induce the changes?
It's a combination of unpredictable maternal separation. So we take the pups one day after they are born, and we separate them from their mom every day for 3 hours, anytime during the day. In addition to this, during these 3 hours of separation, we stress the mother. So anytime during the 3 hours, so it's unpredictable again, we put her on 2 different stressors, one stressor. It's not the same stressor every day. It's random as well. So she's not only stressed by being away from her pups, but she's also stressed physically by being placed either in a cold water or in a tube, restraint tube. So it's really a combination of physiological, emotional, physical stress or trauma. It's quite traumatic for the pups. The mother takes care of her pups when they are reunited, but it's just the maternal care is hectic, it's unreliable, and it's not as good. The quantity is there, but it's not as good quality as it should be. So there is some kind of neglect and poor maternal behaviors.
Are you looking for changes in the pups or the mother?
In the pups, essentially, when they are adult, and in their own pups and the pups of the pups. So that's where the multiple generations are.
But why stress the mother then? Why not just take the pups away and leave the mother alone to hang out, and then you bring them back? Because it seems like that adds in another factor.
Because it's not sufficient to produce symptoms which are severe enough to be transmitted across multiple generations. And, in real life, if we want to be close to human life, violence or hectic families or, traumatic events related to war, to conflicts or very complicated situations, usually it's not just one thing. It's a combination of factors. So that's why we really wanted to recapitulate things which can happen in human.
Yeah, but the problem is, how is the mother feeding the pups? Are they feeding from her? Are they breastfeeding? And if so, she could be producing all kinds of different substances and things that they're consuming that's changing them in a different way than you think.
Oh, yes, absolutely. But it's no problem for us, because what we are interested in is whatever is induced in them, whether this will alter their behavior and physiology, and whether this will be transmitted to the following generations. So it doesn't matter how this is induced.
Can you bottle feed, essentially? The reason I ask is that may be the mechanism that really, it'll be very hard to pick it apart later. What if will pups feed from another mother or not? Will she reject them? And if so, again, can you hand feed them at a certain point? Because if you hand feed them non-stressed milk, that would probably be dramatically different than stressed milk. Maybe it's not just the licking and the grooming behavior. Maybe it's the food they're eating too.
No, you are completely right. It's difficult to do. You cannot bottle feed pups, and feeding in mice it's almost constant. They feed almost all the time. So if we use a control mother to feed them, it will be as if they have been raised by a control mother. That would be, and we would need to separate the mother as well. So that would be a bit too complicated, I think. But yeah, you're right. We checked actually the milk of the stressed mothers, and there are alterations in this milk.
Yeah, so that changes the nutrition. Maybe you could attack it from that angle.
Yeah. Yes.
What changes in the milk, and maybe you could introduce that.
Yeah. That's good. That would be again, that would be a bit difficult, just to feed them with milk from stressed mothers. No, I don't. No one has ever done this.
Yeah.
No, that's a complicated problem.
Well, have you been able to analyze the milk to see what's changed?
Yes, we did. We analyzed the microRNAs in the milk, and we saw that there are changes. Whether this is sufficient, to be leading to the symptoms that we see, perhaps it probably contributes to the metabolic symptoms that we observe, because we see that the glucose regulation is altered. There is a dyslipidemia in these animals. But whether this induces the risk-taking or the depressive phenotype and the antisocial behaviors, I don't know. It could be, but that would be quite surprising.
Well, here's another issue, is that there's probably a microbial constituent to the milk. So what if it's the stress in the mother changes the microbial attachment to the milk, changes the mix of the microbes and their metabolite, and that's also changing the milk?
Yes, that's another possibility. We looked at the microbiome of the pups and the offspring. We did not look at the microbiome of the stressed mothers themselves. But you are right that it could be a contributing factor. But again, the questions that we are asking are not really at this level. They may be at some point, but we are really more interested in how the resulting symptoms, regardless of the way they are induced, regardless of the mechanism of induction, whether we know that's yes, and how they are transmitted across multiple generations.
Okay, yeah. I got you. I'm getting too down into the biology of it, but so what have you noticed so far? What kind of correlations do you see?
Correlations, you mean in between generations, or
Yeah, you said that some will last longer. The risk-seeking behavior lasts a lot longer than the other effects. Are you trying to characterize the intensity of the stress, the duration of the stress? What are some of the factors that you want to piece together and figure out?
No, we are really interested in the mechanism of alteration of germ cells. What happens in germ cells, which explain the transmission across generations. Regarding the effect of different levels of stress, yes, we did this at the very beginning when we established this model almost 20 years ago now. We tried different combinations, predictable separation, unpredictable separation, predictable stress, unpredictable maternal stress, combination or no combination of stress to the pups and stress to the mothers. And really it's the combination of all these parameters, unpredictable maternal separation plus unpredictable maternal stress, which was strong enough to give symptoms which were transmitted to the following generations. If you don't make that unpredictable, for instance, if everything is predictable, happening at the same time of the day, there is no transmission to the following generation. So yeah, we know that a really strong trauma is necessary to induce symptoms which are transmitted.
Are males affected differently than females?
Globally not, but behavior is different in females and males. But globally, most symptoms are observed in both females and males. But for instance, for social behaviors, social behaviors are different in males and females. So the consequences of this trauma is a little bit different. While in females, we see mostly altered social recognition. In males, that will be more antisocial behaviors. While males will be kind of not more aggressive, but they can't really respond properly to aggressive peers. In females, we don't see any change in aggressiveness. But overall, globally, they have the same comparable phenotypes.
What about duration of the effects through generation? How many generational effects are there? Same for men and male and female?
Yes, the only most symptoms. All symptoms are present consistently to the third generation. Some disappear at the fourth, and really again only risk-taking is present to the fifth or the sixth. Metabolic alterations, we have not yet tested them systematically in the third, fourth, and fifth. Yes and no on small cohorts, but they are still present in fourth generation, but not in the fifth anymore.
So there's so much to look at. Here I am. Who am I? I'm giving you advice, but I would think that what is the organ that produces the sperm in mice, for instance? What's it called?
That's testes.
The testes. Okay, testes. So I don't know how many cell types are there in the testes.
Oh, hundreds.
Really? Just in the testes is different? Yep.
Well, it depends because, there is a complex, differentiation of depends what you call cell types. Germ cells, they can be in many different states because they go on differentiation.
I guess one thing you could do to focus is take a particular cell type, in one of these mice and stress it and see if you can sample some of the cells, every day or every week for a time period, and then try to look at least, let's say, their methylation pattern, compare that same cell type, same cell, just different stress points, and see what differences you see, maybe to see if there's tons of changes or just a few, and maybe that would help to start clue you in on areas of the DNA or other areas that are affected.
Yeah, no, it all depends on what you are interested in. We are not interested in the acute effects of stress. We are interested in long-term effects of stress. So we stress the animals when they are pups. We wait weeks and months before checking them. So we really want to make sure that we look only at the things which are persistent and which affect them when they are adult, even if they were induced when they are pups.
I see. Interesting. Are you able to look at, the function of their other organs to see how, so the stress has all essentially been condensed in the germ cells. Now a new generation comes, they carry forward the stresses. Mm-hmm. But I would think that all the cells of their body, all their cell types have been changed, not just their germ cells. Mm-hmm. So maybe somehow you can see differences in organ function in the succeeding generations.
Absolutely. That's what we have seen, actually. I mean, we see that pancreas, the metabolism is quite severely affected. Glucose is dysregulated, insulin also, lipid metabolism is dysregulated. So pancreas, liver, all the metabolic organs, fat, adipose tissue has a number of changes. Bone has many changes as well. Hematopoietic stem cells are altered, so the immune system is affected. So basically everywhere we look, we also looked at skin, and skin innervation is affected. Everywhere we looked, we saw an effect.
Wow, I didn't realize it's that pervasive. It's amazing.
Yeah. Well, yes and no. You imagine that a young individual is exposed to chronic stress for a very critical period during life, in early childhood. This can only have dramatic effects everywhere, right? Because every tissue is being developed. It's a critical period for every organism, human or animals.
Yeah. So what would be a thrilling result for you to figure out over the next few years?
There are two major levels. The first one is really to try to identify all of the major or the most important epigenetic alterations which can be induced persistently by exposure. And so identify the epigenetic factors in germ cells, in particular, both males and females. We've been working on this model since 20 years. So it took us 20 years to be at a stage where we have a solid model, which is reproducible, which has consistent symptoms transmitted across multiple generations, and where we can really start to screen different epigenetic factors consistently across tissues. So it took us all this time to get to this stage. So we would like now to really enlarge the catalog of all the changes that we see in germ cells in males and females. That will take another couple of years, because in particular for male germ cells, because there are all these different stages, from the germ cell, the germ stem cell itself. So there are stem cells, they are called spermatogonia cells, to the mature sperm. So there are many things that we have to look at, how an epigenetic alteration which is induced in the stem cell, how does it evolve, which changes stay, and we believe that there is a kind of a cascade. There is an initial change which is induced by exposure, and it will transfer the signal to another change, which will then transfer to another type of change. So it's really a cascade of events, and we don't really know how this transfer of information, of pathological information, is done in the epigenome in germ cells. The second level of analysis is how these epigenetic alterations are induced in germ cells. what is the link between being exposed to a trauma, for instance, or high-fat diet. Well, we are mostly interested in trauma because it's non-invasive. It's all psychological. It's all driven by the brain. It's more complex, but it's a lot more. It's very relevant to human beings. So, by which mechanism is being exposed to a trauma when you are a child is going to alter the epigenome in your germ cells in a persistent manner to alter your future gametes. So we have started to work on this question a couple of years ago, we postulated that blood is probably the mediator of the signals between the brain, which captures the trauma, the stress, and the germ cells. And we have, very interesting data showing that indeed there are factors in blood, fatty acids in particular, which are significantly altered across generations and which are powerful ligands to receptors which exist on germ cells. So they are perfect candidates to be signaling molecules in between the blood and germ cells.
Couple of quick questions here. So how long do the pups feed or are cared by the mother typically? And then I guess mice live, an average of 3 years or something?
A little less, 2.5 years. And the pups, they are weaned at 21 days, but they start feeding themselves when they are 14, 15 days.
Okay, and then when does the stress stop versus when is the pup going to breed?
The stress is between day 1 to day 14 of life. Then they are weaned, so it's 2 weeks of stress. Then they are weaned. So there is after the 2 weeks of stress, there is 1 week when they are with their mother and there is no stress. Then they are weaned when they are 21 days old. And then we test their behavior and metabolism and so on when they are adult, which is starting at 2.5, 3 months of age.
Well, at what age do they breed? So you can test the next generation.
We breed them generally when they are between 3 and 5 months old.
Okay, and then it's another few days, and then you'll test the next generation of pups at 21 days or 2 months or something like that.
No, also in adulthood. It depends. We have also collected germ cells, for instance, and blood when they are pups, but most of our analyses are done in adulthood because we cannot do everything. It would be too much.
Right, but in general, after the stress has stopped, it's at least a few months, and you're still seeing it, transgenerational effect. Okay, exactly. I got you.
Yeah, that's the point. It's the power of the model. It's really the power of the model because it really shows that there are changes which can really persist.
Yeah, no, it makes sense. I guess it makes sense in people too. If you've had childhood trauma, it can last you your whole life. It can affect your children as well.
Yeah, exactly.
Huh, interesting. All right, so what I guess for people that want to learn more, what's the best way for them to get in contact and look at your research, read papers, that kind of thing?
Well, they can find me in Google. I have a website. My lab has a website with my email. If they need advice on literature, they can email me, or for those who have access to PubMed, all our papers are on PubMed.
Okay, and your last name is Mansuy.
Yes.
Okay, very good. So people can search and find you. Mm-hmm. Any last items that you want to discuss that I haven't asked you?
No, just maybe just the importance of this type of research for human beings and for the concept of heredity, because it really changes the way people look at or think at inheritance or what we can pass to our children, and it's quite unconventional because it goes against the classical view of heredity being only in the genome or in genes. And this type of research shows that it's not the case. It's not new, it's not us who put this hypothesis forward. It's been shown, demonstrated by naturalists already in the 18th, 19th century. Even the Bible has some examples of transmission of life experiences across generations. So it's something which many, many people have observed and which was kind of ignored or even criticized at the time of the domination of the DNA and the old DNA, old genetic decades that we have gone through, which have been quite detrimental for epigenetic, I think, unfortunately. and with all the hope that sequencing the genome will reveal the secret of life, which is completely wrong indeed. So I think epigenetic research has to catch up the delay and the lack of knowledge of people, and the fact that it's not even taught at school. I mean, most people now, it's a bit better. It's getting a bit better these past years, but most people are not really aware. Everyone will know what a chromosome, what a gene is, more or less, but people will not be aware of what epigenetic is, and it's a big lack of knowledge. I think this should be fixed.
Yeah, one last thing to ask you. I don't know. I mean, you probably don't have time or any of that, but have you ever tried to teach any of the rats anything, or the mice anything? How to run mazes and become an expert at it, and see if any learning process gets transmitted to the next generation, whether positive or negative. Any negative association with something, or again, a positive process where they learn something and look for evidence of that in the next generation.
There have people who did this. I think this is, one can consider this possible as long as germ cells will be affected, right? And whether if you learn a poem or a mouse learns, how to solve a maze, is this going to affect the germ cell or send a signal to the germ cell? I don't think so. There is a group, the group of Kerry Ressler a couple of years ago, who published a paper where they took males and they gave them a food shock, and at the same time had them smell a specific odor. And they claim that the progeny and the progeny of the progeny had an increased sensitivity to the odor due to the food shock associated to it. And people claim that it was a memory which was kind of transmitted across generations. I don't know. This study has not been reproduced yet. The data, the statistical analysis in this paper have been quite criticized. I don't know. I can't say yes. I can't say no. I would need to.
I know it's a very speculative area. I was just curious if.
But I think as long as the germ cells are not affected, or I don't think that any learning which happens in the brain is going to influence the germ cells, except if the learning is associated with something which will release stress hormone or something very, very sharp or very abundant or whatever, very adverse, and which will send a signal to the germ cells.
Yeah. Okay. Well, very good. Well, Isabel, it's been good talking to you. I appreciate it. And thank you for coming on the podcast.
My pleasure.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.